So you've already learned about all of the different genetic variety that exists, and that genetic variety, it comes down to the existence of different alleles for a particular gene. Well, what you might not recognize is that all of the variation that exists within living organisms, not just variation between these two lions, not just the variation between you and your siblings or you and I, but all variation among all living things is due to the origin of new genetic variation. So the phenotypic variation is due to the origin of new genetic variation through the process of mutation. If there were no mutations in our species, then all humans would be the same. We would have no variation in eye color, hair color, skin color, height, the abundance of hair, any of those things. And so we are going to explore the process of mutation. And in doing so, it's important that we begin by understanding the way in which mutations are classified or categorized. Okay? So in mutations, we might think first about how they came to be. And mutations might be spontaneous or induced. So a spontaneous mutation is one that simply happens by random chance. And that is certainly the vast majority of mutations that arise within a species. And you can think of this as simply there being an error that occurs during DNA replication. So instead of a G being paired up with a C, it might be a T paired with a C. Okay? That's not stable long term, but we will see a mechanism whereby that transitions to a permanent change. All right, we can also think about mutations as being induced. Okay, induced mutations are those that are the result of exposure to some mutagenic event. Okay, so if you go out and lay out in the sun and get a sunburn, you are exposing yourself to UV light, which, as I've already mentioned briefly in this class, is a mutagen to DNA. Okay, DNA absorbs UV light and it induces mutations, as we will see. Okay, smoking induces mutations. X-rays induce mutations. So spontaneous mutations are those ones that simply happen within the normal daily operations of the cell. I think DNA slippage and the origin of tandem repeat regions in the genome is another spontaneous form of mutation that we've covered already. And then those that are induced are ones that are caused by external factors. Now, the rate at which mutations, spontaneous mutations that is, occur within organisms varies quite dramatically, as you can see here. So bacteria mutate far more rapidly than we do, 100 times more rapidly. And so what these ratios represent in bacteria, 1 in 100 million, okay? So the rate at which a bacterium will accumulate a mistake in their DNA is one mutation, one mistake for every 100 million nucleotides. And I think when I explained this before a moment ago, I might have suggested bacteria mutate more rapidly than us. That's the opposite. So think about it as this ratio. I apologize for misspeaking. For us, for every million nucleotides that we copy during DNA replication or that we synthesize, a mistake is made. In a mouse, there's a mistake every 100,000 nucleotides. Okay? And so these rates of mutation are highly variable among species. And a lot of this has to do with the way in which these organisms are able to repair these types of mistakes. You've already learned about some of the exonuclease activity of DNA polymerases. And a lot of this comes down to those types of cellular enzymes that are able to go in and repair damage to the DNA or mistakes that are made during replication. Bacteria have mechanisms that we have no analog of. They are able to do things we cannot do. All right, now the mutations that occur can also be classified or categorized based on where they occur, okay? And those locations are listed below. I'm going to break them out here. And so genetic mutations may occur in either somatic or germ tissue. Okay, so the cell that suffers a mutation might be a somatic cell or a germ cell. And the easy way to think about these is the germ cells are your gametes. So if it's not an egg or a sperm, it's not a germ cell, and it's in your somatic tissue. Think of your stomach, your skin, your brain, your liver. Those are all examples of somatic tissue. And as you might imagine, the vast majority of mutations happen in these cells because it's simply a random event, and there are many more of these cells replicating. You're not making all that many gametes. But if I suffer a mutation in my skin on my arm as I'm exposed to sunlight, or let's say it's just a random mutation, spontaneous mutation, either way, am I going to pass that mutation on to my offspring? Somatic mutations are not passed to the offspring, right? If I have a tumor somewhere because I had a number of genetic mutations, that tumor is in my tissue. It doesn't get passed to my offspring through my gametes. The mutations that occurred that gave me that tumor occurred in my somatic tissue. We can also think of what type of chromosome these mutations are on, and these two terms, autosome 1X linked, should be familiar to you, but a mutation could be on one of the autosomes. That is, of course, most likely because they're numerically dominant, but X-linked or even Y-linked mutations certainly do arise. Now, when we think about the types of mutations that occur, they can be classified based on their extent. You might remember we talked about chromosomal scale mutations, right? All these translocations and inversions that might occur at the scale of the chromosome. Those are huge changes that occur. What we're talking about now are much finer scale mutations that might be as small as a single substitution of a base, okay? So instead of there being a C at a position, there might be a T. That would be classified as a point mutation. So a point mutation is just one little spot in the genome, there's a change. A missense and nonsense mutation, these are two types of mutations to coding sequence. So these are, by definition, only occurring within genes, okay? And only within the exons of genes. So a missense mutation is a mutation that would be categorized as non-synonymous. And so if you look back at one of the codon charts that we utilized, you can go ahead and Google one of those up, pause the video and look at them. What you will see is if you change, you'll remember from our wobble hypothesis, the codon degeneracy, that if you change a third nucleotide position, frequently that codon will still code for the exact same amino acid. That is a synonymous mutation, that fourth bullet point there, okay? Or a silent mutation. There was a mutation, a nucleotide was changed, but it doesn't have any effect on the product of transcription and translation of that gene because the codon still says basically the same thing, all right? Now, a missense mutation is non-synonymous, and what that would do is change the amino acid that is coded for. So an example of this would be changing the second codon position, the middle of the three nucleotides. If you change that nucleotide from, say, an A to a T, in every instance, it will change the amino acid that is coded for. And you'll remember the example we had in the previous lecture on the difference between the polypeptide coded for by the normal hemoglobin allele and the sickle cell hemoglobin allele. Remember, they differed only in a single amino acid. That is a missense mutation. there was a change to the amino acid structure of the polypeptide, and that results in a change to the function of that protein. Okay, so a non-synonymous change means there's a change, and the after is not the same as before. Okay, a nonsense mutation is one of the few instances where if you change one of the nucleotides and it becomes one of the 3 stop codons, for example, UGA, which is a stop codon, if you change a codon in the middle of a gene to a stop codon, what does that do? That is going to affect not transcription, okay? Remember, transcription only, the RNA polymerase is looking only at the initiation and termination sites of transcription. But if you put a stop codon in the middle there, when the ribosome translates that RNA, it is going to reach a stop codon right in the middle of the mRNA molecule, and it is going to terminate the synthesis of the polypeptide. You will get an incomplete polypeptide, and you will not get a functional protein. This is the type of mutation that has resulted in the O blood type, which we'll talk about again in a little bit. So it's important that you understand when presented with a mutation that is a single nucleotide, that you know whether it is a point mutation, that's a single mutation, but whether it would be classified as missense, nonsense, or silent, non-synonymous, or synonymous. All 5 of those terms should be very familiar to you, and you should be able to identify any mutation I explained to you as being one of those. Okay. Now, the particulars of the nucleotides involved in the mutation will determine whether the mutation is a transition or a transversion. And this has only to do with what type of base has been substituted for what type of base. Okay? And so in a transition, it is a purine that has been substituted with a purine, okay? Or a pyrimidine substituted with a pyrimidine. A transversion is when you have a switch from purine to pyrimidine or the opposite. Okay, I've got an image on the next slide. There's a figure in your text from which I derived this figure. Shows you the difference between transitions and transversions. This makes it clear based on the chemistry of the bases, but you should understand the difference between a transition and transversion and be able to identify them. Mutations might also be lethal, and we've talked a bit about lethal mutations, or at least alleles that are lethal. So yeah, just it should be quite clear that if the genetic information encoded in your chromosomes suffers a mutation that makes inviable an incredibly important gene that produces a critical protein, that it could result in the death of the individual. For transitions and transversions, remember, transition is like with like. So changing a purine with a purine or a pyrimidine with a pyrimidine, and it's useful to view the chemical structure of the bases in order to recognize that a transition is like switching with like, okay? So if you switch from an A to a G at a particular site, that's a transition, okay? Now, a transversion is switching from purine to pyrimidine or pyrimidine to purine, okay? So make sure you recognize those. These are simple classification terms, but you want to be familiar with these for sure, okay? Now, one important last type of mutation that I want to introduce you to, and you may remember this from intro bio, and that is a frameshift mutation. So a frameshift mutation is one in which the number of nucleotides in a coding region, so this only happens in the codons or exons of a protein coding gene. A frameshift mutation affects the reading frame. Remember I told you that the genetic code is commaless, And because there's no punctuation or spaces, that it has to be read 3 letters at a time, codon by codon. Well, because of that restriction, if you delete a nucleotide, for example, this G that's illustrated here, what you do is you change the nucleotide sequence only by one amino acid. Sorry, one nucleotide. You remove one nucleotide, but what it has done is, since the ribosome is going to be reading from that methionine codon every 3 nucleotides, it's now going to read the second and third codons as different than they were originally. They're supposed to code for GLU and ALA, but now that they're slid to the left one position, it has completely changed the codons and what they code for. So a frameshift mutation has effects that go all the way down the mRNA. From that spot on, every codon is affected. And as you can imagine, that can have catastrophic effects on the production of a polypeptide can be folded into a functional protein. So here's an example using the English language and a 3-letter word sentence. And if you take the sentence, "the cat saw the dog," and if you simply do a substitution on the far left, then these have relatively little effect. The bat saw the dog, the cat saw the hog, the cat sat the dog. These do change the meaning of the sentence a bit, but there's still a bit going on that you could interpret, okay? It affects one of the words, not all of them. Now, if there's a loss of a letter, a deletion, this frame shifts every letter to the left by 1. So if that C from cat is lost, instead of "the cat saw the dog," you can see how it changes all of the last 4 words of the sentence, from "cat saw the dog" to "ats, ought, head, og." Okay? This is a frameshift mutation. But what I hadn't made clear to you just yet is the fact that a frameshift mutation can also result from an insertion, the addition of a nucleotide that is not supposed to be there. So if you throw an M in the middle of the word cat, then it throws off in the same fashion every word after that insertion. And similarly, the coding region of a messenger RNA molecule would be completely disrupted. So here's a DNA example in which you can see at the top the original DNA sequence, the original RNA that would result from transcription of that, and then the protein, well, polypeptide, that would be assembled based on that messenger RNA molecule. Now, with a base pair substitution, whether it's a transition or transversion on the left side, you can see that it affects only that first amino acid. Okay, it should be SER, but it goes to either PRO or THR. Now, frameshift mutations, you have an insertion, throws things off. Okay, it changes every single codon and every single amino acid after it. Now they went ahead and threw in a deletion afterwards, which changed things a bit. And it does revert some of those codons back to the way they should be. But from these examples, I hope that you have a pretty clear understanding of the difference between substitutions, these examples being missense substitutions, and frameshift mutations. Okay. So if all of these mutations are the source for all of the diversity of life, then why is it we try to not do things that induce mutations? We put sunscreen on so we don't get UV exposure and get mutations. We should not be smoking because it induces mutations. Why do we try to avoid mutations if that's the source of all awesome genetic variation in the world? Well, Think about it this way. The vast majority of your genome, as we've already learned, is not coding sequence. It's not genes with exons, okay? Most of it is kind of junky stuff, kind of like the repetitive regions we've been learning about quite a bit. So most mutations have no effect. They don't break anything, they don't make anything better, okay? And this is in part because the redundancy of the codon degeneracy model, okay? You can change, these non-coding regions, no problem. All of those are going to be neutral. You can change nucleotides and introns, again, no problem. You can change a lot of third codon positions, no problem. But you change codon position 1 or 2 and we've got some problems, potentially, okay? So the vast majority of our genome is non-functional, and so you might think, okay, well, not only are these mutations not causing me problems, they're also not, they're not making me better, they're not causing any problems, so what's the concern? The issue is that most mutations that do have an effect are going to have a negative effect. And I put it to you this way. Think about your automobile and your reliance upon it for getting to class or getting to work or getting wherever you need to go. And if every time you went out to your car to go somewhere, you just opened the hood and moved some wires around, okay? Because mutations are random, which I've made clear to this point, if you just randomly rearrange things under the hood of your car, which is a finely tuned machine, do you think it's going to run well every time that you go to drive it? Probably not, okay? Most random changes to highly organized things are not beneficial, okay? So that is why we try to avoid subjecting ourselves to mutation. That's why we put on sunscreen and don't smoke and don't subject ourselves to X-ray. Because most of the times, those mutagens are not going to turn us into the Incredible Hulk or the Flash or Captain America, right? Those aren't the kinds of mutations that we're going to be getting. We're going to be getting melanomas and lung cancer, okay? So most have a deleterious effect, but there are so many individuals in every species, and they produce so many offspring, most of which aren't going to survive. That's more common in animals than, wild animals than it is in humans. But most of those offspring aren't going to survive. And so this process of natural selection and evolution weeds out those individuals that have deleterious or bad mutations. But out of the millions of individuals in each species, those few that have those very rare beneficial mutations, those individuals do well, okay? And they have more offspring, and that mutation becomes more common in the population. This is the basis of evolutionary biology, okay? And it's something that I hope you take that class, whether it's with me or Dr. Ballinger, but you can learn a good bit more about how these mutations affect the health and fitness of the organisms that bear them. Now, I'm just going to point out an example of a mutation that has occurred in humans and discuss it a bit in the context of whether these mutations are beneficial or not and what happens to beneficial mutations, okay? So here we can see a phylogeny, a tree that shows how all of these organisms are related to one another. Think of it as a family tree, okay? So a chimpanzee is most closely related to a bonobo, okay? They are sister taxa, they're each other's closest relative. And if you trace the point where their two lines join together, go down from that and you reach a common ancestor with humans and chimps. This is 4 million plus years ago, okay, that we have a common ancestor with chimps and bonobos. And that's how a phylogenetic tree is read. You go further back, and there's a common ancestor with gorilla, further back, orangutans, and way, way, way back, mice, all right? So what we're looking at here is a phylogeny of these organisms as we understand them to be related, right? Chimpanzees are more similar to gorillas. They share a more recent common ancestor than they are to orangutans, okay? Humans are more similar to chimps than they are gorillas, so on and so forth, okay? Now, what we're looking at here is the phylogeny with some information presented about a single gene that is present in all of these species, FOXP2. So this FOXP2 gene, this is a gene that codes for a protein that is very important in the biology of mammals, okay? Mice have it, orangutans have it, gorillas, all of these species have this gene. Now, if you compare all of these organisms to one another, how many mutations do they have that are unique to that species? Okay, so for example, if you compare a chimpanzee and a bonobo, how many synonymous changes have occurred in this gene in those two species? Well, what I see here is that the bonobo has 2 mutations, synonymous mutations that have happened in this gene. So think of those as being third codon position mutations in the FOXP2 gene. The chimpanzee has a single synonymous mutation. So if I compare chimpanzee to bonobo, they have 3 differences, 3 nucleotides that are different in FOXP2. And they are probably all third codon positions, okay? And if I compare a chimpanzee to a human, there are 1, 2, 3, 4, 5 synonymous differences between them, okay? Mutations that don't affect the FOXP2 gene. There are 6 differences between human and bonobo that are synonymous. They don't affect the protein in any way. And what's amazing is between humans and gorillas, there are very few. There are only 3.5. Between humans and orangutans, there are only about 10.5. So these synonymous mutations, they accumulate, but not super fast, okay? Mice are much, much further down the family tree of mammals than the distance between orangutan and mouse would suggest. And so mice are really, really different. And for that reason, they've been separated for a long time from these species, and they have 127.5 on average synonymous changes in this gene. So this gene, lots of mutations happen to it, even in closely related species like the apes, okay? But what is remarkable is look at how many non-synonymous mutations have occurred in these species, in the chimpanzee or the bonobo or the gorilla. Even the orangutan has only 1, okay? But humans have 2 non-synonymous changes. Now, what's been really interesting here and intrigued biologists is that a mutant form of the FOXP2 gene, a mutant allele, is common in individuals that have speech difficulties, leading some scientists to believe that this gene is critical to speech in humans, okay? And the fact that there are 2 non-synonymous changes that exist in all humans, differing from all other apes, tells us something about the origin and the evolution of these mutations, that these mutations are very common in humans for a reason, that these non-synonymous mutations don't happen very often, and when they do, if they're bad, they disappear. That's why there's zeros in chimpanzee and bonobo and gorilla probably. They probably broke the gene if they've had non-synonymous mutations so badly that it didn't survive. Humans have 2 mutations that apparently were beneficial, so beneficial that they now occur in every individual. Okay, if we look at these mutations across all of these apes for another locus, you can see again an example of fixed non-synonymous substitutions that are quite common in humans and suggest selection for this beneficial trait. But you can also see all these variable synonymous sites. All of these green marks on the tree indicate a point where the gene has suffered a mutation, and that mutation doesn't cause any problems, and it has persisted in that species, okay, or in the ancestor of two species. For example, the three green lines that precede chimpanzee and bonobo, those are mutations that happened in their common ancestor, and they have no problems having these mutations. So most have no effect. Some have a deleterious effect. Many of them that do have an effect are deleterious and they're gone. And then very few have a beneficial effect. Okay, so we're going to talk now about human blood groups, and particularly the O blood type, because this is a nonsense mutation. You will remember this image that I showed you that explained the different blood types, A, B, and O. And the fact that O is what it is because it lacks antigens sticking off of the blood cell. Well, the reason that individuals with type O blood have no antigens on the surface of their blood cells is because this is the result of a nonsense mutation. There was a mutation in the middle of the coding region in one of the codons that resulted in the premature termination of translation. You stick a stop codon in the middle of the region of the messenger RNA, and you're not going to translate the rest of it. And for that reason, they make no antigen, and they are type O. Okay, so at the molecular level, we're going to dig into how some of these mutations might arise. Slippage is a great example. This is one that we touched on quite exhaustively when we talked about repeated regions of the genome like microsatellites. I had a great slide that showed you how that newly replicated DNA could sort of fold back and it could continue replication in the wrong spot, creating different sized fragments of DNA. We can also have what is called a tautomeric shift. So what this is, it's the shift of a proton on the base. So one of the bases suffers a shift in the position of one of the protons, and the binding of the nucleic acids, or their binding properties or affinities, shift, okay? And so what you see here is a thymine bound with a guanine, which is not supposed to happen, okay? Or a cytosine binding with an adenine. Again, not supposed to happen. You're not supposed to have hydrogen bonds forming between these different bases. But it happens when the protons shift on one of the bases, causing it to act like something else, causing it to bind or have an affinity for binding with another base that it would not normally bind with, okay? So in this case, the amino form of the cytosine down there and the enol form of the thymine are not binding in ways that they normally would. We can also have what is called depurination. And so this is when the base on one strand is deleted, okay? Now, note this just says base, not the whole nucleotide. The base on one strand is deleted or lost somehow, and it's replaced at random during replication. So that could be problematic, as you might imagine. So the other is deamination, which is a way in which cytosine or adenine, they can again change their binding properties so that the cytosine acts a bit like a uracil, or the adenine becomes something called hypoxanthine, as you see here. Again, these, I'm not going to show you these on an exam. I just want you to know the different types listed on the previous slide that can cause anomalous base pairing or the mutation or change of a nucleotide. Okay, so tautomeric shifts, depurination, deamination. Understand what those are, but again, I'm not going to show you pictures of these molecules and expect you to tell me which it is. Okay, so I want to walk through a tautomeric shift and show you how that can result in a real mutation that is perpetuated throughout the future of that cell line. So we start at the top of this slide with a double-stranded DNA molecule. A is bound to T's, G is bound to C's, 2 or 3 hydrogen bonds respectively. And in the middle, we see that the strands have split. Okay, that's how replication happens. We split apart the double-stranded DNA, and what we have on the right strand is this adenine has undergone a tautomeric shift to the amino form. Now, because replication is semi-conservative, you will see that the left strand goes over to the far left, the right strand goes over to the far right, and we have new strands kind of in the middle there. Okay, and on the left, there's no problem. That left strand didn't suffer any shift. Everything's cool. The new strand that's made, TACG, is correct, is the perfect complement to ATGC. Everything's good. Now on the right, when reading and making a strand complementary to TACG, what the polymerase saw when it hit that adenine immunoform tautomer, it did not read it as an adenine. It reads it as a guanine, basically. And so when it creates a new strand, that new strand reads ACGC. Okay, so now we have on the left a new double-stranded molecule that's A-OK. On the right, we have a new double-stranded molecule that's got a mismatch going on. That's problematic. Okay? So what's interesting is that that tautomer can shift back to its normal amino form. Okay? But in this case, it only does it after that double-stranded molecule splits apart to replicate. So what's over on the left is still a C. Okay? And so the strand to the right shifts back to the normal A, it goes down and it is replicated, and we get a new normal double-stranded molecule that reads just as it should. But on the left, when that strand ACGC is replicated, it's going to be paired up as TGCG. And that's right, that's correct. That G is what should pair up with a C. The cell doesn't know that it should be thinking back 1 or 2 replication steps in time to know that that C over there on the left shouldn't be a C, it should be a T. Okay? There's nothing else for the polymerase to go on. This is not a tautomer of C, it's a real C. Okay? And so now we have, imagine this was mitosis. We have 2 cells. One gets the chromosome on the left, one gets the chromosome on the right. We now have a cell, the one on the left, that has a completely different allele for this locus. And every time it undergoes mitosis for the rest of that cell line and its descendants' lives, it will perpetuate this mutation. It's there for good. Okay, now I'm going to talk to you briefly a bit about how UV light induces mutation. Okay, so if we look at the visible spectrum of light, right, that's a very narrow range in the 750 to 380 nanometer range. And to the left, you'll see there's infrared, microwaves, radio waves. Those are things with high wavelengths and low energy. If you go to the right, you get to UV, X-rays, gamma rays, cosmic rays, right? And so those are the things that we start to worry about. UV, we put sunscreen on. X-rays, when you go to the dentist, they put that big lead vest on you, right? X-rays are not great for you, and the technician frequently leaves the room. Gamma rays and cosmic rays, I don't even want to think about it. I've seen the Fantastic Four and what happens to them when they're exposed to those cosmic rays, so I watch lots of superhero movies. I know what those do. It's bad. Well, it can be good, but for the most part, I think that's probably not realistic. So I'd avoid gamma and cosmic rays. UV light, though, as we've talked about, is absorbed by DNA. It's quite abundant here on our planet. It's high energy, low wavelength, high energy. And what it does when it hits our DNA, it causes thymidine residues to form along the DNA. So it causes these two bases on the exact same strand to bind to one another. That is super wonky. That is not supposed to happen, right? These bases are supposed to be hydrogen bound to the bases on the opposite strand, but UV light screws that up and locks them together. That is a problem, okay? Now, fortunately, there are mechanisms of repair that we are able to fix this problem. So look at this top DNA strand where we have these two thymidine residues attached to one another. They're not bound to the A on the other side. Could be problematic, okay? Now, if that dimer is detected, the surrounding DNA actually kind of opens up, so all those other hydrogen bonds up and downstream of it break as well, and that strand gets cut out, okay? And a DNA polymerase will come in and fill in this gap, getting rid of that thymidine dimer. That is good. That is what we want to happen. But recognize the fact that when you go to the beach and you don't have sunscreen on and you get roasted, you didn't just get one of these and your cells are going to fix it. You probably got loads and loads and loads of these, okay? And you're going to fix most of them. If you only fix most of them every time, that leads to problems over time, okay? All right, so let's think about how bases that are not what they should be are repaired and removed. You know all the players in this DNA repair game, okay? So this is not going to be a very new process for you. So here we've got this unusual U, this uracil, present in the DNA for some reason, and we won't get into why that might happen. Just recognize that sometimes there's stuff in the DNA that shouldn't be in there. It might be due to depurination. It might be due to a number of random things, okay? It might be a tautomer. There are cellular mechanisms for repairing these. This just kind of indicates how, going through a few steps. So there's an enzyme, uracil DNA glycosylase, that finds and removes all of these random uracils that end up in our DNA. And then an endonuclease that recognizes, hey, somebody came here and cut something out. Polymerase comes in, puts a nucleotide there, puts a C there. And then ligase comes in and connects that C to the A, okay? And now it's all repaired. So these mechanisms of repair, the one in which tautomers are cut out and replaced, or a repair like this, are incredibly important for our survival, right? Because we are copying 3 billion nucleotides every time one of our cells goes through mitosis, and the mistakes are happening at a rate of somewhere in the neighborhood of 1 for every million, that's a lot of mistakes, right? How many millions are in your genome, okay? So that's every cell has that many millions, and then multiply it by the number of times your cells have divided. Lots of mistakes. You need to be able to repair these mistakes, okay? And what's interesting is there are a number of conditions, xeroderma pigmentosum being one of them, in which these individuals have a genetic mutation that unfortunately inhibits their ability to actually do these repairs. So you and either of these people go to the beach, you get some UV light exposure, you suffer some mutations, and your cells are going to go in and fix those mutations. These individuals have no repair capability whatsoever. They will not repair any of these mutations that happen to them. And for that reason, they have a 2,000-fold higher rate of the chance of developing cancer than you or I. Okay? So this is a really stark example of the impact of some of these mutations that can happen. And this is the double whammy of a mutation to a gene that is responsible for repairing mutations. Okay. Your text goes into how your cells are able to repair a double-stranded break, because this is potentially very problematic. Okay, this is not something we often think about in the context of the mutation of our DNA, but imagine what happens if one of your chromosomes gets cut or broken. This could be potentially catastrophic. Now, look at this second from the top. You have these two pink portions of this chromosome that were broken, and they actually have kind of some overlapping ends there. That's because this break was identified, and the 5' end of the middle bottom and the top left strand were chewed back. So now I've got some single-stranded DNA hanging off. The reason everything we're about to go through has to happen is because if you walk around and find a stick broken into two pieces lying on the ground, and you pick it up, and it's just got a straight knife cut right through it, you could glue that stick back together, but what you don't know is, was there another third piece that went in between these two? Were there two breaks and that third piece is gone? You don't know if you want to glue these two pink pieces back together that were broken right smack dab in the middle. You don't know if there was something else that was in between and there were actually two breaks. And for that reason, the pink chromosome has to associate with a homologous or identical chromosome. In this case, it's an identical sister chromatid, because we're obviously at some point after the S phase prior to anaphase. Okay? So these two chromosomes, or chromatids in this case, associate with one another and are able to use each other. So the pink chromosome uses the intact blue chromosome in order to fill in the gap and make sure that it fills in everything that is missing. Because if there was a bit stuck in the middle of the two fragments that were discovered, that little bit could be created through the replication that you see taking place with these new purple strands. You would get that little extra bit that's missing in the middle. Okay? And so if, this is a tough thing for me to even attempt to ask you a question on an exam about. I just want you to see the ways in which your understanding of DNA replication can help you grasp the complex mechanisms of DNA repair, whether it's cutting out a single nucleotide and replacing it because it was the wrong thing, or replacing something as dramatic as a completely broken chromosome. And we can use the identical or homologous chromosomes that are in the same cell and use DNA replication to fill in those gaps and then put those chromosomes back together. All right, so now we're going to start thinking a bit more about what causes mutations and what are the consequences of mutations. And when we think first about cause, there are both intrinsic and extrinsic factors that might cause a mutation. And intrinsic factors, you might have, I guess XP is probably not a great example of an intrinsic factor that causes the mutations, but it would allow it to persist. Okay? Now, an extrinsic factor that might cause a mutation could be something like X-ray, exposure to X-rays or radiation. A great example would be, you might have heard of the recent court case in which someone won a judgment against Monsanto saying that they got cancer because of exposure to lots of Roundup, right? That weed killer. So that is arguing that Roundup is an extrinsic factor that induces mutations and caused this person to develop cancer. Okay? Well, how do we actually determine whether things are cancer causing or do cause mutations, right? Why do we worry about exposure to asbestos or secondhand smoke or radiation? What makes us think that those cause mutations? Well, there's actually a very simple test that is used called the Ames test. And the Ames test is still used today. It's very commonly employed to test specific agents for their ability to induce mutations. And so what we have are some auxotrophs, bacterial auxotrophs that are growing in liquid media. And we grow these bacteria and we're going to plate them on so there's liquid culture on the top tubes, and then we're going to plate those on a agar plate, okay? And on the left is a control, okay? And so what we have are these bacteria that are histidine auxotrophs, meaning they can't produce histidine. And so if you put them on a plate with minimal media, they're going to die because they can't produce histidine. You've got to supplement their media with histidine. But if you put them on the left on a plate, you put millions of bacteria on this plate that does not have histidine, what you would expect is you might get a couple individual colonies that grow. These are individuals that just randomly had a mutation that allowed them to again produce histidine. So they're called spontaneous revertants, right? This was a spontaneous mutation. They reverted back to being able to produce histidine. Now on the right side, what we have is a paper disc that we set in the middle of this plate where we're growing all of these bacteria. And this disc has some mixture of whatever the mutagen is. Maybe it's Roundup. Maybe it's, Kool-Aid. I don't know. It's something that somebody is worried might cause mutations, okay? And so you soak this disc in it, you set the disc on the plate, and then you spread a bunch of those bacteria around. And if what you observe is something like you see here, you see lots of histidine revertants growing around that disc, that tells you that being in proximity to that disc and whatever agent is on it caused an increase in the rate of mutation of these bacteria. Because just by chance, you only expect to see, based on our control, 4 revertants. But here, there are so many revertants that it's unlikely that this just happened by chance. It is almost certainly a consequence of exposure to whatever is on that disc. So the Ames test is a great way of determining whether a particular chemical, for example, might cause mutations. And so there are a lot of mutagens that we are aware of, right? And so the things that cause mutations we know are bad for our health, and they might affect us and our offspring. So examples of mutagens that we've talked about: UV light from the sun, smoking, even tanning beds. Gosh, please don't go to tanning beds, right? Go get those amazing, beautiful orange spray tans, okay? You're not going to get cancer from those. But these tanning beds pumping out lots of UV light, you're just absorbing lots of UV light with your DNA is what you're doing. So let's look at an example of a mutagen and an experiment in which the effects of a potential mutagen, in this case polluted air, are tested. So just close your eyes for a second and I'll explain to you the scenario. We are going to raise mice in two different places. One is out in the country at Dr. Noonan's house outside of small town Oxford with, while it's warm and muggy, it's very clean air. Okay? So that's a very rural site. We're also going to raise mice in downtown Dallas. Okay? Also hot and muggy, but there's a whole lot of pollution in the air. Okay? So we are going to raise mice in both of those places, letting them breathe the fresh outdoor air. But we're also in both of those places, at my house and in Dallas, going to raise the mice in an environment in which they're breathing the same air, but it runs through a HEPA filter. So any pollutants or contaminants are removed. Okay. Now look at the chart. Okay. What we have on the left are all the mice that were raised out at Dr. Noonan's house. And on the right are all the mice that are raised in Dallas. After 10 weeks, they examined the rate of mutation in both the paternal and maternal lines. And so what you will see is that at Dr. Noonan's house, the blue and the green bars are about the same height, and those little whiskers on top of them are the error. So they are within error bars of one another. They are very close to one another. They don't differ from one another significantly. The same is true of the maternal lines, the red and the orange for Dr. Noonan's house, right? Now, let's look at the mice from Dallas. What you see is that both the maternal parents in both HEPA filter and no filter, so orange and red, there's a pretty low mutation rate in the females, but in the males we see a very elevated, very different rate of mutation in the males, the paternal line, when their air is not filtered. Way higher. So that blue bar is way higher than the green bar in Dallas on the right. And so what we can infer from this is that there is a mutagenic effect of exposure to this unfiltered air in these urban industrial sites. Okay, so we've talked so far about a lot of different changes that can happen to the DNA, a lot of ways in which the chromosomes can be changed, whether it's a point mutation, single nucleotide that's changed, or wholesale chromosomal inversions and all sorts of large-scale mutations. Okay? What we're going to finish up with is a way in which portions of the genome can actually move around. Okay? And this also has an effect on the organism's health. So what we're talking about are called transposons. And what transposons do is transpose themselves. They move. So these are also frequently called jumping genes. Now there's a fairly lengthy section on transposons in your text, and we are not going to get into quite that much detail, okay? I've given you this example of a transposon below in this slide, but I want you to basically understand that these transposons are highly mobile and they can put themselves damn near anywhere in the genome, okay? And that can be a problem. Now, there are loads and loads of transposons in your genome, thousands of them all over the place. A significant portion of your genome is only there because of transposons. Because 1 became 2, 2 became 4, right? They jump around and duplicate, and they end up all over your genome. Some organisms, like salamanders, which have enormous genomes, way bigger than yours, their genomes are 85% transposons, or mobile elements. So it's all of this junky stuff that has just jumped all around and stuck itself in random places. They are characterized by this internal sequence, and with that internal sequence, they basically carry with them the gene, the instructions for the enzymes that allow them to jump. Okay, so that makes a lot of sense. Let's just look at a very simple example that's presented by your text that explores the potential impacts of these genes. So we're going to talk about a system in which there are simply 3 genes. Here, you are seeing only 2 of them. I'll call it D and W, okay? In the absence of this gene AC, D and W are fine, okay? The gene D works, the gene W works, everything's fine, okay? Now, if there is this other element within the genome, it can cause the transposition of DS, okay? And DS jumps all over the damn place, okay? It might jump in, by chance, right next to W. What that can cause is the breakage of the chromosome. We've talked about Fragile X syndrome. I don't want to get into the details of exactly why the chromosome would break here. I'm just showing you that as things jump around the genome, there can be serious consequences, one of which might be the breakage of the chromosome. DS could, on the other hand, jump in the middle of W, okay? If you stick something in the middle of gene W and you go to transcribe it, you're going to end up with a whole bunch of crap right in the middle of your RNA, your messenger RNA, when you go to transcribe it. It's garbage. W is not going to work. That gene will not be expressed. But that DS gene can also jump right back out of gene W, okay? So the movement of these transposable elements can be very consequential, right? And it can also greatly affect the organization of the genome and even the size of the genome. Okay, that really concludes our discussion of mutations of DNA. We've gone through all the types of mutations. I encourage you to do whatever you feel comfortable doing, whether you're searching YouTube or just Googling for different types of genetic mutations, types of mutations. Make sure that you're comfortable with these different types of mutations, and particularly the synonymous, non-synonymous, missense, nonsense. Those are things you really need to be very comfortable with, okay?